for the introduction, and thank you to Dr. Cameron and colleagues for inviting me here today. I'm going to be talking about some interesting work that I've been doing since I was a pulmonary fellow 25 years ago, and still haven't given up yet. I'm hoping that someday it'll actually lead to fruition. Let's see how we get this moving forward. These are my disclosures. Importantly, I have no specific financial interests in any of the companies that I've collaborated with, but if there are any companies that want to give me financial interest that are in the audience, I'd be glad to accept it. We're going to be talking for the next 15, 20 minutes about a relatively new concept. So you've heard about large-scale pharma-driven clinical trials of mostly small molecules or monoclonal antibodies. I've been working, for good or for bad, for the last 25 years on a concept called gene therapy. So this idea, as it was conceived in the early 1990s, was the idea that if we knew that, for example, a disease like cystic fibrosis had an abnormal gene, that if we go in and correct that genetic abnormality, that we could correct the phenotype, that we could potentially cure the disease by altering the genetic makeup of the target cell. In cancers, we actually have it relatively more simple, which is that we don't necessarily have to correct a single gene. We have to just alter the cell to make it more susceptible to the patient's own environment. So, for example, this is a target cell. This is a mesothelioma cell, could be a lung cancer cell, could be a kidney cancer cell, and you have what's called a vector. A vector is a device that can deliver genetic material to the cell. We've been using adenoviruses, but these can be lentiviruses, they can be nanoparticles, they don't have to be viruses at all. But they can contain genetic material, often DNA, but it can be RNA, it can be siRNA or microRNA, and you deliver it to the cell. There's a receptor for the vector on the surface of the cell, and then it's intercalated within the cell, taken to the nucleus, where the genetic material is then transcribed and then in the cytoplasm translated, and then to produce a protein which will alter the cellular makeup. So, for example, in using adenovirus in mesothelioma, we felt that there were a few advantages for us. So one is for thoracic surgeons, interventional radiologists, interventional pulmonologists, the pleural space is pretty readily accessible. We can put small catheters in, we can put scopes in there, the patients tolerate exceedingly well. You can do a thoracoscopy under local anesthesia on an outpatient basis, and you can deliver whatever your drug of interest, in my case it was a gene through using a vector, into the pleural space. We also got very lucky, which is that mesothelioma cells have on their cell surface what's called CAR, Coxsackie adenovirus receptor. And this receptor is about 10,000-fold higher on mesothelioma cell surface than on normal mesothelium in the pleural space. So when you put adenovirus into the pleural space, there's a relative selectivity for the tumor cells compared to the normal mesothelium. And that was completely serendipitous. And then lastly, we found that the pleural space is such a huge surface area that it's an ideal place to generate anti-tumor immune responses. So as we morph the type of genes that we are delivering into genes that we're going to activate immune responses, it turns out that the pleural space is extremely unique and for doing this. There are a variety of gene therapy approaches that have been studied by us and by others, and very simplistically, you can take tumor out of a patient, genetically modify it, kill the tumor cells so they can't reproduce themselves, and then inject it under the skin as a vaccine. For the most part, these have been done and have failed miserably, although almost every tumor type has been tested with these types of ex vivo vaccines. In situ, the variety of things that you can do with genetic replacement or genetic addition. So one of the first was, well, many tumors have a wild type have a mutation in the p53 gene, which is a tumor suppressor gene. So the idea was, if we had a normal copy of the gene, why not inject it into the tumor cell and see what happened? Well, it worked in correcting some of the tumor cells, but the reality is that you can't get enough of this wild type gene into enough cells to actually affect the patient and improve survival. We've been working on something called suicide gene therapy, which I'm going to talk about. There are a variety of other approaches which Dr. Jahan alluded to, including oncolytic viruses. I actually have spent most of the last 15 years working in transfer of immunostimulatory genes, including type 1 interferons. I'm actually not going to talk about that today, but I'm glad to talk about it over lunch if you want to discuss that further. And then you're going to hear from my former colleague, Emin Moon from Penn, who's going to talk about chimeric antigen receptor T cells, which are autologous T cells which are genetically modified to kill specific tumors. So we've been working for the last 25 years on something called herpes simplex virus thymidine kinase suicide gene therapy. So what is suicide gene therapy? So the idea is they're trying to get the cancer cell to commit suicide, which would be a nice thing to do, leave the patient intact in the process. So here we have a tumor, a cartoon of a tumor, and the suicide gene is here. So this is the genetic material brought to the tumor by a vector, and the suicide gene produces a protein, which is typically an enzyme, and the enzyme by itself is non-toxic. And this non-toxic enzyme will be in a few, but not all, of the tumor cells. You then expose the tumor to a prodrug. Those tumor cells that express the enzyme or have the enzyme in them can convert the prodrug to a toxic metabolite. That toxic metabolite will kill the tumor cells that express the enzyme, but it can also leak between cells that actually have the enzyme and those don't, can kill adjacent tumor cells. This is called the bystander effect. So, for example, we did a lot of experiments when I was in the laboratory 25 years ago, that if 5% of the tumor cells would express your suicide gene and you expose that tumor to the prodrug, you could kill over 90% of the cells in the tumor. So the suicide gene that we're working with was a gene from the herpes virus, the herpes simplex virus thymidine kinase gene. And you put this into a tumor cell through a variety of mechanisms, and then expose that tumor cell to the prodrug. So the prodrug, which HSV-TK converts, is ganciclovir, which is used for CMV infections, or acyclovir and valcyclovir, which is used for herpes virus infections. And what HSV-TK does is it converts it to a phosphorylated form. So normally thymidine in the cell, which is one of the key makeup proteins or makeup molecules of DNA, is converted to a triphosphorylated form, which allows for DNA elongation. But if instead you have ganciclovir and you have this HSV-TK present, this is the rate-limiting step for the conversion of ganciclovir or acyclovir or valcyclovir to a triphosphorylated form, which then disrupts DNA elongation and kills the cell. So back when I was in the laboratory in the early 1990s, we developed a skid mouse model of mesothelioma, taking human mesotheliomas out of patients at the time of surgery, homogenizing those tumors and injecting them into the peritoneal cavities of immunodeficient mice. It could grow tumors that looked like peritoneal mesothelioma. We would then inject them with a single dose of the vector, gave them 2 weeks of ganciclovir, and we could nearly cure these animals of mesothelioma. We then did the same thing with an immunocompetent rat model. This is a rat model where they take asbestos fibers, inject them into the peritoneal cavities of rats, wait about 6 months until the rats develop ascites, harvest the ascites, grow the tumor cells. Now we have a syngeneic model of mesothelioma that you can then put directly into the thoracic cavities of rats. And we could then get dramatic reductions of tumor by, again, a single injection into the pleural space of this AD-HSTK vector with 2 weeks of ganciclovir. And this was the graph that we took to the FDA in 1995 to get the first human gene therapy trial in mesothelioma approved. And this was not very impressive compared to a lot of animal studies that we've done later on or others have done around the world, but we showed that compared to a control, which was a control virus injected into the tumor, that this is the combination of the TK suicide gene and the ganciclovir showing a prolongation of survival in this immunocompetent animal model. So back in 1995, as I said, we got approval by the FDA to do the first human gene therapy trial. This is before PEMSIS. This is before surgery and certainly before lung-sparing surgery. And so we were allowed to treat patients de novo who had never had any treatment before. So this is, for example, a biopsy. All of the patients would go to thoracoscopy. We'd obtain baseline biopsies. We'd place a chest tube. We then waited for the fluid to drain. We injected the vector right through the chest tube and then repeat the thoracoscopy at 72 hours. And these are the biopsies of patients at 72 hours. And this is immunohistochemistry directed against the TK protein. So every red spot you see is this TK protein in a tumor cell. And we were literally high-fiving ourselves in the laboratory. We saw all these red cells on the immunohistochemical stains. And then we did a low power, and we saw that these tumors, which are often 1 centimeter or 2 or more thick, and you heard how large some of these tumors are when they were ablating them, we had maybe 40 to 50 cell layers. So then we realized that this is a superficial transmission of gene, and that although we could have the proof of principle that the gene was getting into the tumor cells, that we were unlikely to kill tumors. But we had forgotten about the bystander effect. And so this is a patient with sarcomatoid mesothelioma. Again, we didn't have a lot of these options in the early to mid-1990s, so this patient had no other treatment. And we had biopsied these tumors and confirmed the presence of sarcomatoid mesothelioma. He had the vector put into his pleural space. got ganciclovir. Day 60 had no evidence of disease in his thorax. This is all inflammatory response to the adenovirus. 2 years later, he died of intra-abdominal progression, but had at autopsy no evidence of intrathoracic disease. We did a series of patients, probably about 40 patients, in a series of phase 1 studies in the late 1990s. And what was really interesting is that we saw some long-term responses, but that the responses that we saw were very delayed. So this is a young woman from Australia who was diagnosed during a pregnancy. She was diagnosed, and they had nothing to offer her in Australia in 1998 when she came to us. We enrolled her in the study, a single dose of the AdTK vector, 2 weeks of ganciclovir, with no other treatment, no chemotherapy, no radiation therapy, no surgical resection, no cryoablation. 8 months later, this is her pleural disease in the right hemithorax. Not much change 8 months later. 5 years later, almost no detectable disease. She lived 14 years, never got any additional treatment. Every 6 to 8 months, she would develop what felt like pleurisy to her. Likely was tumor regrowth, and then she had probably had memory T cell responses from the original AdTK vector that we had put in. So 14-month survival, we all have these kind of anecdotes, but what it told us was that the major effect of this vector was not that we were killing tumor cells by the suicide gene effect, but that we were stimulating anti-tumor immune responses. And so we and others over the last 15 years or so have started to reanalyze our data and to look at what we were actually doing. And of course, give it a cooler name. So instead of called suicide gene therapy, the same approach is now called GMCI, or gene-mediated cytotoxic immunotherapy. And the idea is that the way these vectors, in combination with a prodrug, kill tumor cells, they kill tumors also in such a way that they dramatically increase the local immune response and the presentation of tumor antigen to dendritic cells, and then change the way that T cells see these tumor antigens on dendritic cells in such a way that they dramatically elevate systemic anti-tumor immune responses. And so this is just another way of showing the same thing, that standard approaches, let's say, for example, you had a cytotoxic chemotherapy that itself was not particularly immunogenic. You kill a tumor cell. You may have some tumor antigens, but the environment in which those antigens are being presented is not that immunostimulatory. You then have presentation of those antigens without a lot of co-stimulatory molecules to T cells, and you may even induce immune anergy. So that actually, not only do you not get an immune response, you may get a negative response. You may have increases in regulatory T cells in this setting. With GM-CSI, this is one of the most profound immunogenic cell deaths that you can actually have, and you then stimulate this presentation of tumor antigen to dendritic cells. You convert dendritic cells from immature to mature forms with lots of co-stimulatory molecules to activate CD4 helper and cytotoxic CD8 cells, and you create this systemic anti-tumor response. So this is a review from a few years ago showing a variety of clinical trials in multiple tumor types. This is our study in mesothelioma summarizing all of our data to date. This was from 11 years ago, but prostate cancer, malignant glioma, medulloblastoma, this approach has been used in a lot of tumor types. And right now, the variety of trials using this approach, including a phase 3 randomized trial with surgery in glioblastoma multiforme, a phase 3 trial in hormone refractory prostate cancer, a trial that I'm going to talk to you about pancreatic carcinoma that raises some really interesting ideas about combination approaches, and then a trial that we just presented at ASCO this past June, which is for mesothelioma and lung cancer, ovarian cancer, and pancreatic cancer that had spread to the pleural space. So this is one of the studies from JCO that was presented. This is the same AdTK vector that we were using in mesothelioma that was used in malignant glioma. These are patients who had baseline debulking surgery, so they couldn't be completely resected, and then in the tumor bed had the AdTK vector injected into the tumor bed, and then got 2 weeks of valcyclovir. What you can see is these are the re-resection immunohistochemical stains showing a dramatic increase in T cell infiltration into the tumors. And again, going back to the biomarkers that Dr. Jahan was talking about, one of the biomarkers that we think is going to determine whether patients may or may not respond to immunotherapy is the presence of T cells within the tumor microenvironment. In the laboratory of my former colleagues back at the University of Pennsylvania, they looked at an esophageal carcinoma model and treated patients with the AdTK vector and valcyclovir in advance of doing surgical resection in a syngeneic murine model of esophageal carcinoma. And they showed the same thing that you just saw in the glioma model, which is the AdTK dramatically upregulates T cell infiltration and specifically CD8 T cell within the tumor microenvironment. And a control virus, control adenovirus with LAC-Z does not do that. So this is specific to the expression of HSV-TK and the production of superantigens in the bystander effect. We then took this back to our laboratory when I was still at Penn and did an experiment where we took a lung tumor model. This is a TC1 tumor, which is a syngeneic tumor model of lung cancer in a flank. This is the unchecked growth of controlled tumor in the flank of an immunocompetent mouse. Here you see the effects of chemotherapy, in this case gemcitabine and cisplatin. Here is the effect of the HSV-TK vector and ganciclovir just by itself, but the combination nearly cured all the animals. So the combination of chemotherapy in this approach was very exciting. And based upon that, my colleague Charu Agarwal in thoracic medical oncology at Penn and I designed a phase 1 trial. We took patients with mesothelioma and malignant pleural effusions who were not candidates for surgical resection but were diagnosed de novo. So these are all patients, patients who came in de novo, not all of them. But they had to have a patent pleural space so we could put in an indwelling pleural catheter. We then drained them. We put the vector in once or twice into the pleural space, gave them 2 weeks of oral valcyclovir, and then sent them back to their referring oncologist, and they got whatever chemotherapy would be required based upon the oncologist's choice. If it was lung cancer, they would get whatever the oncologist wanted to give for lung cancer. The primary endpoints were safety and toxicity, determination of maximally tolerated dose, looking for gene transfer in the fluid that we would get from the pleural catheter that we put in, and then secondary, we're looking for immune responses, cytokine responses, and then tumor responses by CT and PET. So we presented this at ASCO as a poster this past June. We showed that we had treated a number of patients in 3 different dosing cohorts. So you dose this by viral particles. So this is a lot of virus. 1 times 10 to the 12 viral particles was cohort 1. Then we went to 1 times 10 to the 13th. And then, as I'll tell you, we then added celecoxib for a variety of reasons into our last dose cohort. And celecoxib actually turns out to be really interesting as a combination therapy, not the least of which is you can walk into your drugstore and just get it if you need it. They all got 2 grams of valcyclovir TID as part of standard dosing for 14 days and then went through standard chemotherapy. Again, this is the schema for the approach. Patients would come in, they'd have their pleural catheter placed. If you were in the 3rd cohort, you started with celecoxib before dosing, but you got your 1 or 2 doses of intrapleural AdVTK, your valcyclovir. Chemotherapy would start on day 14, and then serial CT scans every 6 weeks. So this is the study. We had 19 patients in this phase 1 study. 14 were mesothelioma, 4 non-small cell, 1 breast cancer patient. This is the median ages. The ECOG performance status, you had to be 0 or 1. These are the meso patients that we had included. They were all pleural mesothelioma. The majority were epithelioid, which is the normal distribution. You would see that the majority of patients are epithelioid, but would include some sarcomatoid and biphasics. We did have 4 lung adenocarcinomas. And in terms of the line of treatment, the majority were first line, but in the mesos, we had some second and third line that were included as well. In general, it was pretty well tolerated. We did see some what we call cytokine release syndrome, which is seen in a lot of immunotherapies, fever, chills, sweats, rigors, not dissimilar than what was described in the Listeria trial. And that's why for the 3rd cohort, we had 1 patient in the 2nd cohort who ended up in the ICU with transient hypotension, resolved within 24 hours. But we decided to add celecoxib, which we had used in our interferon alpha 2b gene therapy clinical trials as a therapeutic combination, but it also showed that it mitigated the cytokine release syndrome. So we added celecoxib as a pre-treatment in these patients. We saw no more cytokine release syndrome. And the most common grade 2 abnormality that we saw in laboratories was lymphopenia. So what we saw in this phase 1 trial was very interesting. So we saw in these 17 patients 5 partial responses. 3 of these were in mesothelioma patients, 2 in non-small cells, 8 patients with stable disease, and 4 with disease progression. 7 of the 17 are still alive in an active follow-up at an average follow-up of 5 to 21 months. These are the survivals and the characteristics and the other treatments that the patients received. Here are the PRs that we saw, and most of them were seen at the higher dose levels. This is one of the CT scans from patients. Again, this is a non-controlled study, and all these patients were getting chemotherapy. So I can't tell you whether the response that I'm showing you here is related to the standard chemotherapy they received, to the ADH-VTK, or to the combination. But what we saw, as many people see, is that there is a reduction in the size of tumors in a variety of areas in the hemithorax, pre and post. This is after ADH-VTK and 4 cycles of carbopem in a patient with an epithelioid mesothelioma. So it's interesting, but it doesn't really tell us necessarily that our drug was active. What was interesting was that there was a significant dose relationship that we saw. Again, this is a phase 1 study. I'm hesitant to show any survival curves from phase 1 studies because this is an incredibly heterogeneous patient population. But there is a signal here that we went from the lower dose cohort 1 times 10 to the 12th to the 10 to the 13th, and these are mixed between the celecoxib and the non-celecoxib, that we saw a significant improvement in overall survival and PFS in the patient population. So as we look towards the next approaches for this, we think that the higher dose may be better for studying than the lower dose. So what do we learn from this phase 1 study? We learned that GMCI with this ADTK and valcyclovir can be safely given intrapleurally at high doses in combination with systemic chemotherapy, that you can mitigate the cytokine release syndrome symptoms by giving celecoxib prophylaxis, that the response and survival data are encouraging and really push us to do larger scale randomized trials. And then the question comes up with all these studies, should we combine this with checkpoint inhibition? And this is one of the questions that I wanted to ask Dr. Olewski, which is, do we add checkpoint inhibitors because they're cool and really interesting and we need to blend them in with the other tree because we want to study it? Or is there actually a biological basis for synergy? So we tried to ask that question, is there a rationale for why there actually might be synergy between an anti-PD-1 monoclonal antibody and GMCI? So this goes back to what I alluded to before, which is that this is being studied in a variety of other tumors. So this is a really interesting trial for patients with pancreatic carcinoma, where they did endoscopic ultrasound at baseline and took biopsies of the pancreas. And at the same time that they took the biopsies, they injected the AD-HSVTK directly into the pancreatic tumor using endoscopic ultrasound. I don't know if we can drop the lights a little bit so people can see this. Is it possible to lower the lights a little bit? If everyone falls asleep, it's okay. You can see the picture better. So I don't know if you can see this. This is where the EUS probe sits. This is the pancreatic tumor sitting here. And in the next thing, you can see the needle coming out of the EUS probe directly into the tumor in the pancreas. That's great. Everyone will fall asleep, but at least my pictures look good. Okay. And what they did was they took these biopsies, and everyone then went on to surgery. So you had these patients who were surgically resectable. They injected the ADH-VTK. They got 2 weeks of valcyclovir. So you had pre and post-tumor. You had the initial EUS biopsies, and then you had the tumor resection. And they looked at PD-L1 expression in the tumors and CD4 and CD8 T cell infiltration in the tumors. And what they saw was, pre compared to post, that ADTK valcyclovir dramatically upregulates PD-L1 on tumor cells, but it also does something perhaps even more important, which is it dramatically increases CD8 T cell infiltration into the tumor microenvironment. So we don't know what the best biomarkers are for nivolumab or pembrolizumab. We think that PD-L1 expression may be important. Maybe it has to do with the antibodies we're using. We don't know. And we think that having what's called a T cell inflamed phenotype may also be important, which is that having these cytotoxic T cells infiltrating into the tumor. And then the combination itself may be the best sign of all. And what we have here potentially is a way to take a tumor which is not responsive to immunotherapy and prime it so that it may be responsive to immunotherapy. So going back to the data that had been talked about before, mesotheliomas, you're awake? Sorry. This is the data that's been presented, but it's actually really important. Mesotheliomas, some of them present PD-L1 at high levels. If they do, it's bad because they have a poor prognosis. But it may be also that even though it's bad, it makes them more susceptible to checkpoint inhibition. And this was a paper that from human pathology from the French group, which showed the same thing, overall survival negatively correlated with PD-L1 expression. But again, may mean that mesothelioma, if primed by increasing T cell infiltration and PD-L1, may then allow us to have more effective immunotherapy than just in about 20% of the population. So we went back to the laboratory and started to do some small-scale experiments to see if the combination of anti-CTK and anti-PD-1 would improve survival. So this is the combination here. Small numbers of mice. We didn't really characterize PD-L1 expression in our murine tumors, which we need to do. But the company that makes the vector that I use now, which I have no financial interest in, has done this in multiple tumor types and has shown synergy and has shown that T-cell infiltration and upregulation of PD-L1 with AdTK on multiple tumors. So this question of synergy seems to be borne out, at least in multiple murine models. So we go back to the idea about the fact that immunotherapy likely will need to be a cocktail of events, not just one drug. So for it to be successful, we're going to need to generate T cells through vaccines, through chemotherapy, maybe through cryoablation, maybe through adoptive T cell therapy, which we're going to hear about. And then we need to have the brakes here that we can release. So maybe we need to upregulate the brakes a little bit to make them more susceptible. And then we're going to need to bring those T cells into the tumor. And GMCI allows us to bring these T cells into the tumor and may also upregulate these here to make them more susceptible to T cell therapies with anti-PD-1. So I am a small cog in a large machine at my former institution, University of Pennsylvania, and at NYU, where I have just recently moved. And there are too many people to note who I've collaborated with, but mostly my mentor, Steve L. Belda, and the nurses who really